This presentation will cover the most common methods of introducing foreign DNA into your cell culture. This process is typically transfection. This is the question that your experiment will demonstrate if it works perfectly. How do we get mammalian cells to express a desired product? We need to introduce the desired gene and get it to be used as instructions to build a protein, a process called gene expression. A cloning vector is used for this purpose. The vector is a self-replicating DNA molecule that you can insert your gene of interest into. The vector may be a plasmid, a bacteriophage, which is a virus that infects bacteria, or an artificial chromosome that yeasts will use. The vector and the cell, to be the host of your gene, depends on your experimental purpose. The size of your DNA insert and what the inserted DNA needs to do will play into the selection of your system. Artificial chromosomes can take much larger DNA inserts than a plasmid or a viral vector. One of the most important considerations is whether you need the protein to be produced in the system and if the gene originally came from a prokaryotic cell or a eukaryotic cell. Do you remember that prokaryotic genes are more simple in structure than eukaryotic genes? Eukaryotic genes have introns that must be spliced out before the messenger RNA can be translated into protein. This is not true of prokaryotes. Additionally, eukaryotic genes may produce proteins that need modification in eukaryotic organelles to become functional. Therefore, sometimes a eukaryotic system is needed to produce the protein. Getting proper expression of genes in a eukaryotic system is more complex than in bacterial systems, but it is necessary at times, and it is the basis of many new biological therapies on the market today. It's possible to get bacteria to produce proteins from eukaryotic cells, but the gene must be in the processed form. To do this, messenger RNA for the gene is isolated and reverse transcribed into DNA by a viral enzyme. Then it is called cDNA. This is inserted into a bacterial cell vector. So yes, it can be done, but sometimes the protein produced by something like E. coli doesn't work correctly, hence the need to have the protein made in a eukaryotic cell. Vectors need to have as part of their DNA sequence a selectable marker. What this is a gene that allows for the identification of the cells that have been transformed, meaning have incorporated the vector. The most common type of selectable marker is drug resistance. The vector will encode for an enzyme that can inactivate the drug. Cells that cannot inactivate the drug die, and those that have the vector live. This is an easy way to look for the transformants and is used with bacterial plasmids almost universally. The other type of marker provides the cells the ability to live in media without a nutrient that the non-transformed cells must be provided. Thus, if you grow the transformation mix in the medium without the nutrient, only the transformed cells will live. Both of these methods are positive selection because the cells that grow are positive for the vector. Negative selection comes into play when the transformed cells can no longer do something that the non-transformed cells can't. This type of selection is much more time-consuming and difficult. You need to grow the transformation on two types of medium and look for colonies that are missing. This figure shows a type of positive selection. Media without the antibiotic allows all the bacterial cells to grow. Medium with the antibiotic allows only the transformed cells to grow. This figure shows negative selection. In this case, the cells must be provided tryptophan and amino acid if they're transformed. Colonies of bacteria are grown and then plated on medium with and without the amino acid nutrient. Then you look for the colonies that grow with the nutrient, but not without the nutrient. You can see that it requires more effort to identify these transformants by negative selection, but some types of research need this methodology. Vectors do have different properties that are important under specific conditions. We already mentioned plasmid, bacteriophage, and the artificial chromosomes. BAC is a bacterial artificial chromosome, which is used less often than a yeast artificial chromosome, a YAC. A shuttle vector is a vector that will replicate in more than one type of host, or can be used in more than one type of host. Our transfection experiment uses this type of vector. The vector acts as a plasmid and bacteria and replicates, but then it can be used to express genes in eukaryotic mammalian cells. Remember, an expression vector is one that allows the inserted gene to be transcribed and translated, and that's because it has the necessary controls such as a promoter upstream of the gene. These are used when the protein produced is the desired end product. Lastly, vectors may be present within a cell as a single colony copy or as many hundreds of copies per cell. This is important because sometimes overexpression of a protein is toxic to a cell. The researcher must balance getting a large output of the desired protein from the culture with keeping the cells alive and producing the protein of interest. This may require testing several host vectors for your DNA insert to find one that works well in your system. If protein is your desired product, then there are a few common organisms to choose from. It's always best to use systems that many others have used before you because many of the common problems will have solutions described in the literature, so you don't have to reinvent the wheel for every scenario. A very common mammalian cell line is the CHO, or Chinese hamster ovary cell line. This cell line is common in industry. The bread yeast, Saccharomyces cerevisiae, is also a very common expression system, but it does not always work with proteins produced by human cells. Hence, the hamster cell line, which is mammal, like humans, and often will produce functional proteins from human genes. Plants are sometimes used, and one of the most common plants for this work is the tobacco plant. Finally, E. coli is a very common laboratory workhorse. Most often, work with a gene starts in the E. coli and migrates to another system if functionality is not achieved in the bacterial system. It's important to understand how foreign DNA is introduced into a cell. The terms can be a little confusing, so try to keep these straight. Transfection is the term for introducing a gene or other biomolecule into a cell line derived from multicellular eukaryotic organism. Typically, this is a mammalian cell line, but it might be a plant or insect line as well. Transformation is the process of introducing DNA usually, but sometimes other biomolecules as well, into a bacterium or yeast. Transduction is the process of using a modified virus to introduce DNA or RNA into a cultured cell type. There are two common methods of transfection. The first uses modified lipid to form these little vesicles called liposomes. The liposomes form around the molecule of interest, such as a vector. The liposomes fuse with the cell membrane and dump the contents into the cell. This is actually the method behind the RNA vaccines against the virus that caused COVID, the Pfizer and Moderna vaccines. The RNA enters our cell via a type of liposome fusion. Then our cells make the viral protein encoded by the RNA, and this stimulates our immune system to react. The RNA is pretty short-lived, and our cells stop producing the protein, hence the need for a booster to ramp up the response so it's protective against the infection. The second type of transfection uses electricity to punch holes into a cell membrane. The DNA molecules in the surrounding solution leak into the cell before the cell repairs the holes. This method requires specialized equipment. There are many factors that can affect the success of your transfection. The most important factors are the cell type you choose, the concentration of your reagents, the ratio of your reagents, the presence of contaminants that may exert toxicity on your cells, and the storage of your reagents that could affect the potency. Also important are the health and growth state of your cells. Remember that log phase is the best phase to work with cells. Also, the medium you are using. These methods require the removal of serum from the surroundings. Serum has many proteins that can interfere with the reagents. Your method is also important. Did you mix it well? Did you do each step for the appropriate length of time? Add the correct volumes? Less important is the size of your plate. Is it the 6, 12, or 24 well? This is going to depend on your experimental design and the volume of your reagents. Also less important is the medium and the serum used to grow the cells after the procedure is complete. One of the reasons it's good to use cell lines used by others in the past is that you will know if the cells are easy or very difficult to achieve transfection. Always try the most common transfection procedures first before moving on to less common and more tricky procedures. It will be necessary to try various conditions and use healthy cells to find optimal conditions to achieve the desired result. Transfection is a very useful methodology when working with cell lines, but it is a bit tricky to master. I hope our experiment works.